Hello everyone, welcome to my channel Love of Biology. Today we are going to learn about electron transport chain in respiration. Most of the ATP generated during the aerobic respiration of the glucose is not directly they are generated from these pathways. Rather it is derived from a process that begins with moving the electrons through a series of electron transporters by the redox reactions, that is the electron transport chain. This causes hydrogen ions to accumulate in the matrix space. Therefore the concentration gradients forms in which hydrogen ions diffuses out of the matrix by passing through ATP synthetase, and they will synthesize the ATP. They will produce the energy by the catalytic action of this ATP synthetase enzyme, which phosphorylates ADP into ATP. So ATP will generated. So the electrons from where they are coming? They are coming from the substrates, respiratory substrates, by breaking down them. So the electrons will be formed to the oxygen through a series of carriers. The electron transport chain is the last component of the aerobic respiration and is only part of the glucose metabolism that uses oxygen. Oxygen continuously diffuses into the plant. In animals, it enters the body through the respiratory system. In cells, so normally this is taking place takes place in the inner mitochondrial membrane of the eukaryotes, but in prokaryotes mitochondria is absent, so it takes place in the plasma membrane. So next, complexes. There are about 5 different complexes represent that accept or transfer electrons through a chain. Complex 1, 2, 3, 4 and 5. Along with these 2 carriers are present. One is the ubiquinone and another one is the cytochrome C. Let us discuss one by one. First one is the complex 1 or mitochondrial complex 1. It is the electron transport chain is formed of NADH dehydrogenases and the iron sulfur centers. Catalyzes the transfer of 2 electrons from NADH to the ubiquinone, as well as 4 protons will be generated from through the membrane. They will cross the matrix region. So NADH is first oxidized to NAD plus and it will transfer the electrons. So next, ubiquinone. So the electrons from the complex 1 will be transferred to the ubiquinone. Ubiquinone, it is an electron carrier in the respiratory chain in that it is not bound attached to a protein. This allows the molecule to move between the flavoproteins and the cytochromes, and it will transfer the electrons to the next complex, that is the complex 2, which is the smallest one. It is a succinic dehydrogenase FAD and FES centers. Catalyze the transfer of electrons from other donors like fatty acid and glycerol 3-phosphate to the ubiquinone through the FAD and FES centers. This complex runs parallel and it is the smallest one. Next, it will transfer to the ubiquinone, and from the ubiquinone the electrons will transfer to the complex 3. It is consists of cytochrome B, C and a specific FES center. Then enzyme cytochrome reductase will reduce the electrons. It will accept the electrons from the ubiquinone and it will transfer to the next complex, that is the 4. So protons will be transferred to will release across the membrane, aiding to the proton gradient. So the ubiquinone is oxidized back to the reduced form, while the iron center in the cytochromes is reduced to the Fe2 plus, the reduced form. So next, complex 4, it consists of cytochrome A and A3, which is also called as oxidized cytochrome oxide. This is the last complex of the chain and it involves the transfer of 2 electrons from cytochrome C to the molecular oxygen. So the molecular oxygen, it is the last one, and it will react with the protons inside the mitochondria and they will form the molecular water. Next one is the chemiosmosis. So this is ATP synthesis. Actually in the oxidative phosphorylation, the hydrogen proton which are inside in the region will enter the ATP synthetase enzyme and they will synthesize as the ATP. The protons will enter into the ATP synthetase, which have the F0 and F1 region. F0 will act as a proton channel as well as F1 will act as a catalytic site. So the ATP here synthesizes by the ATP synthetase enzyme. The process we call it as a chemiosmosis enzyme. Chemiosmosis. The potential energy stored in the hydrogen ion gradient to add the phosphate to the ADP and forms the ATP. So this, it allows the protons to pass through the membrane and uses the free energy difference to phosphorylate adenosine diphosphate, making the ATP. So these components are important. Thank you for watching. Please subscribe my channel.